Welcome back, guys. Today we are discussing under this topic of vitamins, hematopoietic vitamins. These hematopoietic vitamins are very clinically important. So let's start the discussion. These hematopoietic vitamins are of two types, but they are two in numbers. First one is folic acid. First one is folic acid. This folic acid, as we know, this is the vitamin B9. Folic acid. This is vitamin B9. This folic acid is derived from this word folium. Folium, which exclusively means green leafy vegetables. It exclusively means green leafy vegetables. This folium means green leafy vegetables. So this folic acid is exclusively derived from green leafy vegetables. Green leafy vegetables, that is, it is of plant origin. Bracket, right? Exclusively. So this we have seen, this folic acid. Second we are going to see is cobalamin. Cobalamin is our vitamin B12. So this vitamin B9 and B12 are our hematopoietic vitamins. Vitamin B12 or cobalamin. This is mostly animal origin. This is mostly of animal origin. In bracket you can also have exclusively animal origin. B12. This B9 is exclusively plant origin, green leafy vegetables. This folic acid. So from here we draw a conclusion that the strict vegans, they develop B12 deficiency. The strict vegans develop B12 deficiency. What are the very grave consequences we will discuss in further. For now, remember, strict vegans develop the B12 deficiency. Why? Because these are mostly of animal origins, and vegans, they don't consume animal meat or animal products. So first one we are going to see here, start the discussion, is folic acid or vitamin B9. Commonly we call this as folate. Straight away we are going to dig into the functions of folic acid. So for that we are going to see the active form. This active form of folic acid. Active form of folic acid is THFA. This THFA is tetrahydrofolic acid. THFA, tetrahydrofolic acid. This is the active form of B9 or the folic acid. This THFA is very, very important, a carrier of one carbon group compounds. It's a carrier of one carbon group of compounds for their metabolism. So what are this one carbon group of compounds? We are going to see some examples. Remember, first is methyl. Very simple. Methyl. Next is methylene. Methylene. Third is methynyl. Methynyl. Let me write once again. Methynyl. Methynyl. Fourth one, remember, is formyl. And the next one to remember is forminino. Forminino. So these 5 are the most important. This is enough. Methyl, then methylene, methynyl, formyl, and forminino. These are the 1 carbon group of compounds, and the carrier is THFA, tetrahydrofolic acid, which is the active form of vitamin B9. So we are going to see a very important set of reactions, how this 1 carbon metabolism is carried out. First, we start from this sulfur containing amino acid serine. This is the sulfur containing amino acid serine. Serine here is converted to glycine. Another amino acid, not sulfur containing, but this is another amino acid, and the enzyme here for this conversion is serine hydroxyl methyl transferase. Serine hydroxyl methyl transferase, conversion of serine to glycine. This part exclusively is considered as the, remember, entry point. So the entry point to 1 carbon metabolism. This conversion is the entry point to 1 carbon metabolism. So further what happens is, with this reaction there occurs, so we've seen this enzyme is methyl transferase. So methyl group is transferred for the creation of this compound. Remember, methylene THF, bracket write CH2, methylene THF. Along this reaction, methyl transferase, after the transfer of methyl group, forms methylene THF. Remember, THF, not THFA. These are very important. You cannot interchange words, otherwise it will be conceptually wrong. So this is formation of methylene THF. Then this glycine further gets converted to other compounds. We need not to discuss, remember, because this 1 carbon metabolism is very long. We need to omit this. We are going to discuss only the important parts here. This glycine is getting converted to other compounds. For this methylene THF, which is CH2, is getting converted to methyl THFA. See, THFA is written here, not THF, methyl THFA. Methylene THF to methylene THFA. Methyl THFA is CH3. So on this conversion of methylene THF, this methylene THF is interchangeably converted to methynyl THF, tetrahydrofolate, methynyl tetrahydrofolate, bracket write CH double bond. It's interchangeably converted with methynyl THF. This methynyl THF is interchangeably converted with this formyl THF. Remember, we are dealing with THF. THF, THF. But this here is methyl THFA. Why? We will discuss further. There lies an important concept there. For now, focus on this. It's methynyl THF. It's interchangeably converted with formyl THF. Also, this methynyl THF, it's interchangeably converted with forminino THF. Forminino THF. Again, THF, tetrahydrofolic acid. But the difference here is forminino THF is THFA, tetrahydrofolic acid. It's forminino THFA. We're going to see this forminino THF, CHNH. This is the chemical, and this formyl THF is, this formyl group is CHO. This formyl CHO. Now, one important thing to discuss here: from where this forminino THFA and formyl THFA are coming. Okay, so remember, the donor for forminino THF is histidine. This amino acid is the donor of forminino THF. And what is the donor of formyl THF? Remember, it's important: tryptophan. Histidine and tryptophan are the donors of forminino and formyl THF. Forminino THFA, formyl THF. This we need to remember. We have seen above this methylene THFA. When this glycine is getting converted to other compounds, this is conformed to methyl THFA. This methyl THFA enters. This methyl THFA enters a cycle of reactions where this methyl B12 and B12 are interchangeably converted. Methyl B12 and B12 are interchangeably converted. How this B12 is getting converted to methyl B12? This methyl THFA from this methyl gets separated or gets detached, combines with this B12 and form methyl B12, and they are converted interchangeably. So as this methyl THFA gets detached, the methyl parts get detached, this THFA gets released freely. This is very important. This THFA gets released freely. It's a very important phase in this one carbon metabolism. This THFA gets released. Why? Because the methyl gets detached and forms this B12 to methyl B12. This THFA freely released THFA. This interchange conversion, as we know, is called our folate trap. This methyl THF is CH3, methyl getting detached, converting B12 to methyl, and THFA getting free released. This active form of B9, free released. This entire conversion is called folate trap. For this folate trap, we needed to discuss all this one carbon metabolism drama: this methylene THF, methynyl THF, forminino THFA, and formyl THF. This folate trap we need to discuss here. So that's why this THFA is 10B9 is considered to be a carrier of one carbon metabolism group. After this folate trap, this B12 to methylene B12 is actually involved in this reaction of homocysteine homocysteine to methionine. Homocysteine to methionine. This B12 is involved in this conversion reaction, and we need not go too deep into this. We'll ultimately discuss everything in the amino acid metabolism. For now, remember, this conversion homocysteine to methionine requires this B12 to methyl B12 conversion, interconversion, and this whole scheme of things required for this folate trap. To remember all of this, it's a very feels like a very long reaction. If you revise 2 or 3 times and get the concept, it's very easy. So we have seen here THFA gets freely released THFA. What happens if THFA is not freely released? This THFA is not freely released in free form. From methyl THFA is not freely released. There is obviously deficiency of THFA. There is deficiency of THFA, and all metabolic functions of 1 carbon metabolism is halted. All metabolic functions of 1 carbon metabolism is halted. THFA is not freely released. These metabolic functions of 1 carbon metabolism is halted. So we have seen all this methylene THF, methynyl THF. Now we are going to discuss. We have seen this serine to glycine conversion by serine hydroxymethyl transferase is the entry point to 1 carbon metabolism. We are going to see the exit point. What is this exit point? This exit point is our methylene THF. This methylene THF we have discussed is the exit point of 1 carbon metabolism. Methylene THF, this CH2. This is going for TMP synthesis. This TMP synthesis is a very important metabolic phase in the nucleotide metabolism. For this time, remember, for TMP synthesis. If this exit point methylene THF goes for TMP synthesis, this TMP synthesis is affected. Why this TMP synthesis is affected? Because instead of methyl THFA getting released, methylene THF gets released at this exit point of 1 carbon metabolism. This TMP synthesis is affected, which leads to DNA synthesis getting affected. DNA synthesis is affected as DNA, deoxyribonucleic acid, the nucleic acid metabolism is affected, TMP synthesis is affected, so DNA synthesis is affected. Also, another exit point is our formal THF. Formal THF, it is our CHO. Formal THF is required for going to purine synthesis. This purine synthesis, as this formal THF goes, instead of methylene THFA, purine synthesis is affected. Purine synthesis is affected, and ultimately, as we know, DNA synthesis is affected. This DNA composed of purines and pyrimidines, this purine synthesis gets affected of this formal THFA getting exit instead of this methyl THFA, from which THFA gets freely released. Instead, this methylene THF and formalin THF gets released at the exit point, and TMP synthesis and purine synthesis are affected, which ultimately leads to both DNA synthesis getting affected in both the cases. This leads to the DNA maturation. This DNA maturation getting affected. This DNA maturation getting affected. It's a very important conclusion we get. DNA maturation getting affected. So now we're going to see the all important deficiency manifestations. Deficiency manifestations. First one we're going to see is due to, we've seen here, DNA maturation getting affected due to decreased DNA synthesis. Decreased DNA synthesis due to decreased TMP and decreased purine in the nucleotide metabolisms. Decreased TMP and decreased purine leading to defective DNA maturation. Defective DNA maturation or nuclear maturation. Defective nuclear maturation, which ultimately leads to this condition, megaloblastic anemia. Very, very important. As the DNA maturation, nuclear maturation getting affected, there is megaloblastic anemia, or to be precise, macrocytic anemia. Why to be precise macrocytic anemia? We will discuss later in the B12 when we discuss B12, the cobalamin, cyanocobalamin. To be precise, it's called macrocytic anemia due to deficiency of B9. Then third we're going to see are neural tube defects. Neural tube defects during maturation of embryo and development of fetus. Neural tube defects during maturation of embryo and development of fetus due to decreased DNA synthesis. Decreased DNA synthesis leading to neural tube defects, leading to these two important conditions: spina bifida and anencephaly. Spina bifida and anencephaly. Neural tube defects due to B9 deficiency. So pregnant women are advised to consume B9 supplements properly, as this neural tube defects in the baby due to B9 deficiency. So pregnant women are advised the supplement of B9 in their diet should be proper, otherwise it will lead to decreased DNA synthesis, leading to some horrible conditions such as spina bifida and anencephaly. Then next one we're going to see is this low folate. Low folate can cause cancer. Why? We've seen B9 is responsible for DNA maturation, so any abnormalities in DNA maturation can cause some cancer. For example, colorectal cancer. Low folate can cause cancers like colorectal cancer. So these are all we have seen some of the important deficiency manifestations in B9. Most important is this macrocytic anemia. Megaloblastic anemia don't call such in B9 deficiency. CY precise to macrocytic anemia. Then sphenobypedine anencephaly in fetus and colorectal cancer. Why? Just due to this effective DNA maturation or nuclear maturation, decreased TMP and purine. This is the basis of all deficiency manifestations due to B9. So, an important question is often asked that why folate deficiency is there even if normal diet contains ample amount of folic acid. Why folate deficiency is there even if our normal diet contains ample amount of folic acid? The reason is that there is this enzyme called folyl polyglutamate. Folyl polyglutamates are sensitive to heating during the cooking of food. It's 5 to 10 minutes of heat can destroy 95% of folates. 5 to 10 minutes of cooking in the heat can destroy 95% of the folate. These folyl polyglutamates are sensitive to heating. That's why even if the normal diet contains ample amount of folic acid, there occurs some folate deficiency. So it is sometimes advised to take a bit more than the amount of the folate required. So you have seen. Now we are going to see the biochemical assay. Biochemical assay of B9 folate. Biochemical assay of folate. It's a very important discussion. See carefully. First is the serum folate and the red cell folate. For red cell I mean RBC. This is the assay of serum folate and RBC folate. Next is this histidine load test. Histidine load test. How is this histidine load test is carried out? We have this important compound called FIGLU. For only remember this name, this FIGLU. We'll discuss more in the histidine metabolism. For now remember this FIGLU is converted to glue. FIGLU is converted to glue for conversion of THF tetrahydrofolate to forminino THFA. This THF tetrahydrofolate is converted to forminino tetrahydrofolic acid. It's FIGLU converted to glue. This FIGLU is excreted in urine in deficiency of B9. This deficiency of folate B9, this FIGLU is excreted in urine, and this is the basis of biochemical assay regarding histidine load test. We'll learn more about this FIGLU and glue in histidine metabolism and amino acids, don't worry. For now remember this FIGLU is excreted in urine in deficiency of B9. Third is ICAR excretion. This ICAR excretion ICAR stands for amide imidazole amide imidazole carboxamide, amide imidazole carboxamide, ribose 5 phosphate, amide imidazole carboxamide ribose 5 phosphate, ICAR. This I is involved in urine synthesis. For this urine synthesis, you have seen above, THFA is required. THFA is required for urine synthesis. THFA is required. So this ICAR is excreted in urine in B9 deficiency. B9 deficiency, this ICAR involved in urine synthesis is excreted. The fourth one we are going to see is serum homocysteine. Serum homocysteine. This increased homocysteine in blood causes MI, is myocardial infarction. This increased homocysteine in the blood causes myocardial infarction. Why there is increased homocysteine in the blood? We have seen above here in this range of reactions. This conversion of methyl B12 to B12 and the interconversion in this Pauling trap, homocysteine is converted to methionine. So if this B9 deficiency, in which ultimately B12 metabolism is also affected, B12 to methyl B12 is not converted, there is increased homocysteine in the blood which causes myocardial infarction. We will learn in detail about this homocysteine metabolism, everything in the amino acid metabolism. For now, remember this one. The fifth one, probably the most important one I'm going to see is the peripheral smear. Peripheral smear. Peripheral smear of blood, I'm going to see macrocyte red cells. The RBCs will be larger than usual. Macrocyte red cells. There will be matured eosinophils. The eosinophils will be matured than normal. This will form the teardrop cells. The teardrop cells is classical finding in the peripheral smear due to matured eosinophils, macrocytic red cells. The third one is hypersegmented neutrophils. There is this hypersegmented neutrophils. Neutrophils on the development, the process of development, segmented. Number of segments usually ranges from 1 to 6. So this hypersegmented neutrophils, the segments may reach from 8 to 10 segments. So this hypersegmented neutrophils is a very classical clinical finding in the peripheral smear of patients affected with B9 deficiency. The last one is anisopoikilocytes. Anisopoikilocytes. So these four are the important clinical findings of B9 deficiency. These macrocyte red cells, matured eosinophils, that are the classical teardrop cells. Then there is hypersegmented neutrophils and anisopoikilocytes. All these are due to defect in the DNA or nuclear maturation. The last topic we are going to see is folinic acid. There is this compound folinic acid. This is 5 formyl THFA. Sfolinic acid is 5 formyl THFA. There is this drug called leucovorin. Leucovorin. This is a racemic compound. Leucovorin, it's a racemic compound to combat toxicity of MTX. It's a toxic compound. Toxicity of MTX or other DHFR inhibitors. So MTX is a DHFR inhibitor. For now, just remember the names. This toxicity of MTX or other DHFR inhibitors. There is decreased DHFA and it acts as antifolate. MTX or DHFA inhibitors act as antifolate and they decrease the THFA formation. So with this, we somehow conclude our 10B9 discussion. Very important, very important vitamin to be discussed. The next hematopoietic vitamin we are going to see is vitamin B12, or as we know, cyanocobalamin. So 10B9 is also cyanocobalamin. We are going to see the ring structure. There is 4 pyrrole rings. 4 pyrrole rings. These 4 pyrrole rings are coordinated with cobalt. These 4 pyrrole rings coordinated with cobalt is also called as corrin. CORRIN ring. Corrin ring. These 4 pyrrole rings coordinated with cobalt contains 4.35% of cobalt in one ring. There is 4.35% of cobalt right here. Cobalt in one ring. It's also called cyanocobalamin is also called extrinsic factor of Cassel. Extrinsic factor of Cassel. Vitamin B12 or cyanocobalamin is called extrinsic factor of Cassel. So next we are going to see is the metabolism and absorption of B12. It's an important discussion. Metabolism and absorption of B12. Very important discussion. Listen carefully. When we take food in the mouth, there is 1% passive absorption. 1% passive absorption where in the mouth. In the mouth there is this food coming in contact with saliva from the salivary gland. This food, in presence of this cobalamin binding protein Cobalamin binding protein is present in food. These proteins are given names such as hepatocorrin, hepatocorrin, or cobalophyllin, cobalophyllin, or simple R binders. So different names. Not to remember all this, just remember cobalamin binding protein is food. Cobalamin binding protein goes to downwards towards the stomach. Here in the stomach, see here, stomach, these are the parietal cells. We know parietal cells are in the stomach. Is parietal cells secrete this important compound called intrinsic factor of Cassel. Secret is intrinsic factor of Cassel. This is the intrinsic factor of Cassel is released or secreted from these parietal cells of stomach. Parietal cells of stomach. From where? From body and fundus. From the body and fundus of stomach, parietal cells secrete intrinsic factor of Cassel. This food comes down, binded with cobalamin binding protein. Food. There is also this secretion of pepsin. Parietal cells also secrete pepsin along with the intrinsic factor of Cassel. This pepsin, further down when we go into the duodenum, the duodenum, pancreatic juice enzymes digests cobalophyllin, where you've seen this above. It is one of the cobalamin binding protein. Cobalophyllin are these pancreatic juice enzymes are proteases, important powerful proteases that digest the cobalophyllin. After in the duodenum digestion of cobalophyllin, they come down in the ileum of small intestine. In the ileum there is a receptor. There is a receptor called intrinsic factor receptor. Since this intrinsic factor of Cassel released from parietal cells of stomach, this is a receptor called intrinsic factor receptor. There is a special name given to this, remember, cubulin. Intrinsic factor receptor is called cubulin. This cubulin helps this cubulin itself binds to cobalamin. This cobalamin was free after the digestion of cobalophyllin by the pancreatic juice enzymes. This cubulin in the ileum, which is the intrinsic factor receptor, binds to cobalamin and is taken into intestinal cells. Finally, with all the help of these receptors and binding proteins, above from here, from mouth till stomach to duodenum and ileum, this cobalamin, this cyanocobalamin, the help of this cubulin protein is taken into the intestinal cells. It is finally absorbed After absorption, in blood, it is getting transported in blood, transported through this important compound, through transcobalamin, cobalamin 2, very important compound, transcobalamin 2. Squalene and transcobalamin 2 are very important. In the blood, it is transported through transcobalamin 2 and finally taken to target organs. Taken to the target organs. So this is all about the metabolism and absorption of B12. In the mouth, there is 1% passive absorption, not much. In the mouth, saliva released from salivary glands contains these cobalamin binding proteins, hepatocorrin, cobalamin and arbiters. Remember this cobalamin. After the binding to this, in the stomach, the parietal cells of stomach release intrinsic factor of Castle, which then combines with the food. It is already combined with this cobalamin binding protein. It also releases the pepsin, parietal cells of stomach. It goes down in the duodenum with pancreatic juice. Enzyme digests the cobalamin. The cobalamin binding protein secreted from salivary cells, powerful proteases digest the cobalamin. Now the cyanocobalamin in the food is not free, it is exactly bound to the intrinsic factor of Castle. In the ileum, there are intrinsic factor receptors. Most important is tubulin. It binds to the cobalamin and is taken into the intestinal cells. Without this binding, this B12 can't be taken into intestinal cells. After absorption in the blood, transported through transcobalamin 2, very important compound, is finally taken to the target organs. So after absorption, we are going to see that generally vitamin B complexes are not stored in the body. Generally, vitamin B complexes are not stored in the body, but B12 is an exception. But B12 is an very exception. Where it is stored? It is stored in the liver. Stored in the liver cells as, remember, ADO B12 form. This ADO B12 form stored in the liver cells in combination with transcorin. Stored in the liver cells as ADO B12 form. Generally, the vitamin B complexes are not stored in the body, but B12 is an obvious exception. We have seen about this metabolism, absorption and storage. Next, we are going to see are the functions of B12. Very important. See carefully. The active form of B12, the active form is the group of enzymes called cobamide enzymes. The active form is the cobamide enzymes. This active form cobamide has two types of functions here. First, we are going to see here is methyl B12. This methyl B12, how it is formed, we have seen before. Methyl THFA to free THFA. This THFA is free released and this B12 is converted to methyl B12. This is how methyl B12 is formed. This methyl B12 takes part in the conversion of this. We've also seen before homocysteine to methionine. Homocysteine to methionine. The enzyme involved here is very important. See here, methionine synthase. Methionine is being synthesized, so methionine synthase, or the more technical is homocysteine methyl transferase. From the name you can see homocysteine methyl transferase, that a methyl is getting transferred. Which methyl? This methyl B12 is getting converted to B12, and further on during this reaction, homocysteine to methionine, enzyme is methionine synthase or homocysteine methyl transferase. Very important one, so remember this. Next one I'm going to see is this enzyme is this stored form in the liver cells. We have seen this ADO-B12 form, or adenosyl B12, ADO-B12 or adenosyl B12. We're going to see how it participates in the reaction. We have L-methyl malonyl coenzyme A. Cell methyl malonyl coenzyme is getting converted to succinyl coenzyme A. L-methyl malonyl coenzyme is getting converted to succinyl coenzyme A. The enzyme required here is C, methyl malonyl coenzyme A, methyl malonyl coenzyme A mutase. Methyl malonyl coenzyme A mutase. This requires ADO-B12 form. Specific ADO-B12 form is required by methyl malonyl coenzyme A mutase. Remember this. It's required for conversion of L-methyl malonyl CoA to succinyl CoA. So in this function of B12, we have seen the active form, that is the cobamide group of enzymes. These two, methyl B12 and ADO-B12. Methyl B12 involved in homocysteine methionine conversion, and ADO-B12 is involved in L-methyl malonyl CoA to succinyl CoA conversion. Both the enzymes required serves this coenzyme as B12, ADO-B12 and methyl B12 respectively. Now we are going to see all important vitamin B12 deficiency. What happens if there are vitamin B12 deficiency? So the causes, the first we are going to see is nutritional causes. We have seen starting of this discussion that the pure vegans, as the vitamin B12 source is exclusively animal source, the nutritional deficiency occur in the pure vegans. Next is, it can be through malabsorption. Causes are autoimmune gastritis, or more commonly known as pernicious anemia. Excuse me for the handwriting. So pernicious anemia or autoimmune gastritis. Here parietal cells are affected. Leads to intrinsic factor of Castle affected. It's release of intrinsic factor of Kessel, which is very important in the absorption of this B12 from the parietal cells. These are affected in autoimmune gastritis leading to pernicious anemia. This is a cause for malabsorption. Then the third example is gastric causes. Gastric causes may be parietal cells may be defective, or there may be cancer, or whatever there will be gastric causes in the stomach, the parietal cells. Fourth we are going to see are intestinal causes. We have seen in the duodenum and ileum very important functions of absorption takes place in this vitamin B12. So if there are intestinal problems, there occurs B12 deficiency, especially ileum. Due to ileal resection and stagnant loop syndrome. These two are very important words. Ileal resection and stagnant loop syndromes. Remember this. These are the intestinal causes for malabsorption or deficiency of B12. The fifth one is this very peculiar fish tapeworm. Worm. There is this worm called fish tapeworm. This is called Diphyllobathmium lactum. It is found in Scandinavian countries. Very cold European countries, this tapeworm is found in fish. Consumption of this tapeworm can lead to B12 deficiency. How? Because it has a special affinity to B12. This tapeworm found in Scandinavian countries in the fish has a special affinity to B12 and gets attached to them and causes malabsorption and hence deficiency of B12. So we've seen these various causes, these nutritional causes in the pure vegans, malabsorption due to pernicious anemia, gastric causes and intestinal causes, and fish tapeworm. Next we are going to see the all important manifestations. First is the megaloblastic anemia. Highlight this. Yeah, as I have said in the B9 discussion, the deficiency symptom we tend to call the B9. Most accurately, we call that macrocytic anemia. Why? Because this megaloblastic anemia is reserved for B12. This B12 deficiency results in this megaloblastic anemia. Next is homocystinemia. Why? Because in the homocysteine to methionine conversion, B12 is required. So B12 deficiency manifestation is to homocystinemia. This is a myocardial infarction risk factor. You already seen this before. This homocystinemia is homocysteine increased levels in the blood can lead to myocardial infarction, MI. So myocardial risk factor. Third one, very important, methylmalonic aciduria. You have seen above here the functions. Adobutryl form is responsible for conversion of L-methylmalonyl coenzyme A. This deficiency causes methylmalonic aciduria. Next we are going to discuss very important question, that is why neurological complications in B12 deficiency. Why there are neurological complications in B12 deficiency, but we didn't see any neurological complication in B9 deficiency. So see here, this B12 has two forms we have seen above, the ado B12 form and the methyl B12. The group of cobamide enzymes required for act as the coenzyme for important enzymes in the amino acid metabolisms respectively, ado B12 and methyl B12 forms. This methyl B12 is required for this enzyme methionine synthase. This methyl B12 is required for this methionine synthase. So now we see this set of reactions we are going to see. Listen carefully. There is this propionyl coenzyme A, which is converted to D-methyl malonyl coenzyme A. This D form of methyl malonyl coenzyme A from propionyl CoA. This is further converted to alpha methyl malonyl coenzyme A. This D form is converted to alpha form. This alpha form is converted to succinyl coenzyme A. Here a very important enzyme is involved, methyl malonyl coenzyme A mutase. Seen this enzyme also above. This mutase enzyme, methyl malonyl coenzyme A, converts alpha form to the succinyl coenzyme A. This requires B12. This requires B12. So naturally, due to B12 deficiency, this reaction doesn't occur properly. This reaction doesn't occur properly. So this leads to methyl malonic acid accumulation. Methyl malonic acid accumulation. Which form doesn't matter at this later discussion. Just remember, deficiency leads to methyl malonic acid accumulation. This methyl malonic acid accumulation led to propionyl coenzyme A accumulation. Propionyl coenzyme accumulation. This propionyl coenzyme A, remember, is an abnormal fatty acid. Abnormal fatty acid accumulation. As this propionyl coenzyme A is getting converted to succinyl CoA, which is a raw material for the formation of heme. Propionyl CoA gets accumulated, which is a abnormal fatty acid. Abnormal fatty acid accumulation. So accumulation, and it incorporates to neuronal lipids. Incorporates to the neuronal lipids. Very, very important. Abnormal fatty acid accumulation incorporating into neuronal lipids. This will eventually lead to myelin breakdown. This will eventually leads to myelin breakdown, and this cause subacute combined degeneration. Subacute combined degeneration. myelin breakdown due to incorporation of abnormal fatty acids into neuronal lipids causes subacute combined degeneration. So this is why there is neurological complications in B12 deficiency. This neurological complication used to differentiate between B9 and B12 deficiency. In B9 there is no neurological deficiency, but in B12 deficiency, after diagnosing the peripheral smear, we'll see afterwards, peripheral smear appearance in B12 deficiency and B9 deficiency are almost equal. So to differentiate what is the actual cause, we will see these neurological complications. So next we are going to see the assays of vitamin B12. Assays of B12. There is serum cobalamin, very simple. There is serum homocysteine. There is urine homocysteine. Then there is very important serum methyl malonyl acid, serum cobalamin, serum homocysteine, urine homocysteine, and serum methyl malonyl. This one is the most important. This will be asked in the question, remember. This one is the most important, serum methyl malonyl acid. Further assays are there. There is Schilling test, Schilling tests. Then there is obviously peripheral smear. Peripheral smear. What are the findings in the peripheral smear? There is megaloblastic red cells. There is megaloblastic red cells. There is hypersegmented neutrophils. As we've seen above in the B9 discussion, megaloblastic red cells. There it was macrocytic red cells. Here it is megaloblastic red cells. Then there is hypersegmented neutrophils and matured eosinophils. Matured eosinophils. You can also do some bone marrow cytology. Bone marrow cytology can also be done to assess the nuclear maturation. So this is all about hematopoietic vitamins. Discussed B9, B12. Very important discussion. Very clinically relevant discussion. Lots of questions are asked. Remember everything that I've taught. The difference how to diagnose B9 and B12. The biochemical assays are very important. Remember them. So please do revise them. Thank you guys. Watch you guys in the next one. Good luck.